Good afternoon, everybody. Here from New York, I'm Dr. Dennis Melendez, Cientifico Latino's program director, and I'm very happy to have you here today for our webinar on how to get bright scientific grants. This workshop is being recorded and is part of our CSMI alumni program webinar series. So I'd like to take a moment just to introduce our workshop facilitator today. Dr. Christian Cazares, an immigrant from Mexico, is a postdoctoral research fellow in Dr. Bradley Wojcik's lab at UC San Diego. He uses electrophysiology to bridge human EEG, neural organoids, and mouse models to study neurodevelopmental conditions. Dr. Cazares also co-administers the COBE-KIDF Scholars Program in collaboration with the UC San Diego Hadley Institute for Brain and Mind, which offers a paid summer research experience to undergraduates from historically marginalized and excluded backgrounds in neuroscience. Dr. Cazares has extensive trainee grant writing experience with successful grants, including the NSF-GRFP, the DSPAN F99 K00, the Burroughs Wellcome Fund PDEP, and the UC San Diego Chancellor's Postdoctoral Fellowship. Over to you, Christian. Thank you.
Okay, so if you can maybe put in the chat like what sort of fellowship or funding opportunity you might be interested in applying to like this cycle or maybe in the future. And the reason I ask for that is so that I can like better get an idea of how to like, adjust what I'm saying based on what most people are interested in. I'm also switching screens because the monitor that I use or I was using is kind of blown out because I use it for like a visual stimulus task. And so I'm just gonna like, whenever I don't use it for that, I use it for, what's it called? My own personal research. All right, GRFP, NSF, GRFP, a lot of GRFP. Okay, internal grants, F31. All right, so I've also realized that whenever I go full screen, I need to switch what screen I'm on. So, okay, internal grants, great. So that's interesting, mostly because one thing that you're going to learn across your journey in grant writing is that all grants are generally different formatting, but also the same expectations. And having, been awarded like both NIH style, private foundation, and internal. I mean, it's crazy that I have all three examples right here. One of the most annoying things is figuring out how to say what's required in the vastly different formats that they want you to say them in. NIH style, which is mostly what I'm going to focus a lot of my examples on, has a lot more extensive application. There's like a million different documents that you have to submit. The NSF-GRFP is like what, like 4 pages total nowadays, plus your letters of recommendation. And then the morely internal grants, you're going to find, people are going to want you to write a 1-page research proposal, a 4-page research proposal, and you're kind of going to go crazy with trying to figure out how to put the same ideas in very different page requirements. But a lot of the advice I'm going to give today vastly applies to like all of these different opportunities. And so a couple disclaimers before I start. Also, I'm kind of like, I have a little bit of a list going on right now because I have Invisalign and I'm pretty new to it. So I'm sorry if it sounds like I'm like, have like a huge list. So a lot of what I'm going to be presenting today is based on the NIH style format. And the reason I picked it over the NSF-GRFP is because the NIH style is going to be a little more thorough and explicit into what is expected. Like the NSF-GRFP, you don't need to have like a specific aims page, but there's some more emphasis on these like two very broad categories called like broader impacts and intellectual merit. All of this information overlaps. It's just called different things. And even though you don't have a specific aims page in your NSF-GRFP, the order in which you say things in a specific aims page at the NIH style grant format is very similar to how you should be talking about things before you get into the experimental like design and expected outcomes of your NSF-GRFP. Internal grants often do require like a specific aims format. And I think a lot of that is because people are very used to seeing a specific aims format in academic research, especially in the biomedical sciences, which is where I'm coming in from. So again, another disclaimer is that all the things I'm saying come from the perspective of a neuroscientist. If you're a physical, sciences person, then it's going to vary a lot. Maybe you're going to be more inclined towards the NSF format. So disclaimer on that. I'm just offering one perspective. There's no one way to win these things. The key is to basically ask a lot of people to read it. And there are multiple ways to succeed, which is why you're going to get very different feedback. Some people are going to be like, oh, your background should be this long. Some people are going to be like, oh, you should have 3 aims instead of 2. Trust me, like all of it is going to vary depending on who you ask. And so it's up to you to pick the best compromise. Also, you don't need to be awarded a training grant. I know it's going to sound like a little bit of survivor bias coming from me, but like the vast majority of my peers, some of them who are incredibly more successful than I am, did not get the NSF-GRFP, do not have a graduate fellowship. And as postdocs, they're doing tremendously, especially with like private foundation grants. So you don't need one. So don't feel bad if you, for whatever reason, apply these cycles and aren't awarded, especially how tough things are getting nowadays. A lot of the information that I'm sourcing from are some sources that I used to learn how to write this. So there's going to be a lot of repeated language, specifically through Kate Watson's lab at UCLA. She's a system neuroscientist that I'm acquainted with, and so she wrote like a lab's version of how to write a strong NRSA. I'm going to take a lot of that information from there, as well as like this web resource called Biosciences Writers, the anatomy of the specific aims page. And honestly, this is like the resource I use for writing a specific aims page and teaching it specifically. All of these links are at the end of my slides, which I'll be sending over to Cientifico Latino so that they get disseminated to everybody that attended. So don't worry if you like miss any details. And with that, if you're not fully aware, right, there's a different variety of different trainee-focused research grants. So these are just like some of the NIH versions. The NSF also has like pre-doc and postdoc, as well as the NSF-GRFP. But this is just to get you an idea of something that it took a long while for me to learn what it meant, what the different codes for the different grants mean. So for a lot of you, you're going to be applying maybe to the F31 mechanism. There's both like the parent one, and then there's like the diversity one, which are both evaluated in the same pool, just to like dispel the myth that they're evaluated differently. But nonetheless, it kind of seems like nowadays I would highly recommend you apply to the parent mechanism because you never know what's going to happen. And even if the the diversity version comes back, I would still recommend you apply to the parent mechanism, mostly because, again, you never know what's going to happen, as we've seen. And so, as I mentioned earlier, there's going to be a long list of different documents you're going to need to be submitting for one of these NIH-style grants, which, again, comparing to the NSF-GRFP is huge. So the one thing that I'm going to be focusing on mostly today is like this specific aims page, which I believe, through the different trainings that I've received, is one of the most critical components of this application. Trust me when I say like all of this, out of all of this, probably the thing that gets read the most is this one page. Maybe your research strategy to like confirm some of the assumptions people make about your specific aims. And then because a lot of these are training grants, there's the research training grant plan section as well, which is about like 6 pages that also gets read and evaluated. Because at the end of the day, like a lot of these training grants, it's not necessarily the science that gets funded, it's you as a trainee and your potential to be able to succeed in what you propose. And so the process for the NSF-GRFP is definitely different, but I'm going to go over the NIH one first, and then I'm going to give you my impression of the NSF-GRFP. So when you submit an NIH-style grant, it goes over to a program officer for the institute that you are seeking funding from. So for example, if you study like, I don't know, depression, you might want to submit to the National Institute of Mental Health. Then your grant essentially, given the institute you submitted to, gets assigned to a program officer. So these are the people that are sort of like in charge of determining the direction of a lot of the different research programs within the NIH. Then it gets assigned to a study section, which is essentially like your judge committee or the people who are going to evaluate your grant. So these are usually PIs from different labs across the nation. They're usually NIH funded, and they're generally in the area that your proposal is in. Generally. And the reason I say that is because you only get 3 reviewers to evaluate your application, and it might just be that because your grant is about depression and using mouse models, you might get a reviewer that like maybe studies depression, but using like human fMRI. And that's again, look at the draw, which you're gonna see is probably like 50% of the ability to get a grant is almost just luck nowadays. Then if you score really well in the top 50 percentile, then those are actually discussed in person at the study section meeting with all the different investigators that are part of that study section. The rest are sort of not discussed, and that just means it's a flat out like rejection. But then for the ones that do get discussed, there's one reviewer that essentially vouches for your grant. And so there's one person that introduces it to everybody, and then the second and third reviewer that are part of that group that reviewed your grant also kind of fill in the blanks and sort of give their opinion about why they gave the scores they gave you. And then they give their final scores together, and then everybody votes on the score range that um, those reviewers sort of gave out. And then it goes, here's the thing that I didn't learn till later, right? Let's say your study section believes your grant is awesome and they give like really well sort of scores that determine that maybe you'll get granted in terms of probability. Those are not the people that determine whether or not you get the funds. Then it goes to like the advisory council, which is a higher level sort of with the NIH that determines funding. And those people are the ones that are like, okay, based on all the ones that receive favorable scores, these are the ones that we're going to fund. And those, the number of which ones get funded are highly determined by the amount of funds that are available. So you might have heard terms like pay line. You might hear terms like, oh, are you like, 30 percentile, 20 percentile? All of that means is that out of all the grants submitted, you might score like maybe in the 10 percentile, so like in the top 10% of all grants. But for whatever reason, the funding of that year is determined to only fund the top 8%. Even with a really high score, you might not be selected for funding. There are exceptions to this rule, and those exceptions that I've heard about often apply to trainee grants. So even if you're like below the pay line, you might not get funded, and if you're above, you might get funded. Again, I guess a lot of this is determined by like how people woke up feeling that day, and that's kind of the reality of like a lot of grant writing evaluation. And so the things that people are going to look for, regardless of where you submit. So the NSF-GRFP, you might be familiar. You get 3 reviewers. They evaluate your application based on like how you address intellectual merit and broader impacts. And then those scores get aggregated. And then there also is sort of like the reviewers who give the score and somebody who determines what percentile of all NSF-GRFPs get awarded. But one thing that you might have heard about recently is that now who gets funded in the NSF-GRFP is also highly determined by like the research agenda of the NSF, very overtly, especially this life cycle. So that means that as like if you have a really good application in the life sciences, if for whatever reason the current leadership it really cares a lot about AI, your grant might be viewed a lot less favorably compared to like an AI grant. And that's just kind of the sad reality of the NSF right now. Again, these are all, not the norm. So, but at the same time, it's like, what other choice do we have other than adapt? So I guess what I'm trying to say that, follow the money in what best way you can in terms of like the topics that you research. People are going to want to be able to advocate for your grant. So you need to be very clear in sort of the fundamentals of what makes a grant, right? Like, do you have a testable hypothesis? Are the aims that you're proposing independent from one another? This is a common pitfall that I see from young trainees, that they propose an aim one that the result of will determine what happens in aim two. it's not impossible to get funded that way, but I highly recommend against doing that because, personal opinion, I think it's better science to be able to ask questions or address a hypothesis in a way that if your aim one doesn't work, you still are able to proceed forward. Um, and then one of the things that they're going to want to see, especially with these trainee grants, is like, is what you're proposing actually within the scope of like your PI's research? Because they don't want to give, you know, uh, hundreds of thousands of dollars to train somebody that ultimately is going to be like on their own to train themselves to be able to conduct this research. So one of the things I would say for both the NSF and the NIH is like make sure that your research isn't too wildly different from the people that are like vouching for you, whether it is your letters of recommendation or if you're applying to the NSF as a first or second year grad student that are in line with the aims of your PIs, like for example, R1 or whatever other grant that might have. And then one thing that I've learned that is very critical is like put yourself in the shoes of the reviewer because in the NIH style, there's literally somebody that has to present what you propose. Try to make it as easy as possible for them to like scan the page and find the critical components. So one thing I've heard is like imagine somebody literally being picked on and to explain your grant and then being able to like in one sentence they'll be like, well, this grant is about this. They're gonna ask this question and they'll test this hypothesis to address that question, and they'll do this by using this technique. Like that is how quick somebody should be able to explain your grant. And I know that it's very hard at this stage to think about like a really condensed version of what you're proposing, which is why I highly recommend other people to read your grant and ask them, like, can you give a one sentence summary of what I'm proposing if you were to present this in front of other people? If you get a sentence back that doesn't match at all with what you intended to say, then that's kind of time for you to start simplifying the language in your grant and simplifying how you're setting it up. One of the things that I also learned much later in my career is this thing called the NIH Reporter, the link here. So the NIH Reporter is a database of all successful grants within the NIH. I don't know if the NSF has something like this. I'd be shocked if it doesn't. Um, but essentially, the utility of this tool is for you to be able to search the different years as well as the different mechanisms. So for example, if you're like, I'm applying to an F-31, my topic is on, I don't know, Tourette's syndrome. What are the chances that, this institute, like the NIMH, would care about Tourette's and fund Tourette's versus like NINDS, which is like neurological disorders and stroke? You can literally use NIH Reporter to check the history of like the last, for example, 10 years of funding for the F-31 for both institutes and just keyword search like which one has more mentions of something related to Tourette's or which one has more mentions of the techniques that you're proposing in your research. This is one of the most helpful tools that I found to determine what institute you're going to submit it to. Because you don't want to like, even if technically Tourette's falls under both NIMH and NINDS, you don't want to like submit it to the one that, has maybe two grants ever funded through the F-31 mechanisms versus one that maybe has like five or six. And this is just an example. Um, I'm going to quickly look to see, okay, chat questions. Okay, audio is clear, I hope. I'm getting conflicting answers, but that's okay. How do we know which topics are in season? AI, for example. I know sometimes they're posted depending on the organization, but it's not clear on how to find this information. Um, it can be very clear if you look at the right links. So for example, the NIH has like funding priorities. So if you Google, NIMH or NINDS funding priorities, you will go to, it will give you a page that literally states what the mission of each institute is. And I highly recommend you do this each and every single year that you're going to submit a grant because these things change year by year. So for example, like I'm an autism researcher. Let's just say that the funding priorities for autism are much different this year than they are last year, because of the people in charge. Um, for the NSF-GRFP, it's the same thing. They have funding priorities that you can literally just keyword search, like what is the NSF funding priorities at the moment. Um, the reason I say AI is kind of one of them right now is because even like when I was applying to the NSF-GRFP, it became very clear that there was like a reduction in the number of awards being given to like biomedical sciences and psychological sciences compared to like some of the more computational and sort of like physical sciences. But this information's out there. You just need to like, look it up online. And I'll probably, when I send these slides to Cientifico Latino, I'll give you some examples of those links. Yeah, no worries. So feasibility is another thing that I see people struggle with. It's like, you know, NSF-GRFP, honestly, you can, maybe this is not advice, but I've rarely seen evidence to the contrary. You can propose some really wild things, as long as they're like on paper, really sound and rigorous, and you're covering your bases. And that's because for the NSF-GRFP, before you apply as a grad student, or when you're applying before you're a grad student, you don't have to do the research like at all, um, or even provide like progress reports for the research that you did. So really, kind of, you can get really creative. I wouldn't necessarily like, you know, propose inventing a technique that somebody who is familiar with the field might realize that it's not going to take 1 year. But at the same time, if you have an experiment that it's kind of very hard to like do within like a year, but nonetheless, you don't want to tell them that, oh, um, I'm only gonna do this with like 10 mice because of how long it takes for me to do. Like you want to tell them like, no, I'm gonna do my rigorous controls. I'm gonna do a power analysis, and through that power analysis, I determined that I need like 20 mouse for each genotype for each sex combination. So you can give them like something that, technically might be hard to do within that year, but nonetheless shows that you're thinking about these controls. And that's a little more forgiving than like you coming up with like a new technology, or doing a project that, if you're doing fMRI and you're doing a longitudinal study, might be hard to like propose something that, takes 2 years to do, and only doing the analysis in the last 6 months when all data is collected. I would recommend that, like, think about what you can do. Sometimes people think about like three aims or three main objectives for their grants. Try to think if you can cut it down to two, because the space you free up by doing two aims instead of three, um, will add a lot of clarity to the things that you want to do in those two aims. Like it's better to do less and explain it a lot more clearly than just hit them with everything that you think you can do because you want to show off, how, I don't know, how great you are at like doing research. And then, very importantly, like whoever writes your letter of recommendations, if you do propose a project that might be like, oh, interpreted as ambitious, you want to make sure that your PI is somebody who can like very directly in their application, say that there's evidence supporting your ability to conduct this research. Like one of the things could be like, oh, they're already learning how to do this technique in the lab, or like they've already shown that they can, carry out a project, in a similar way within a year. Basically, like have your letter writers address that very directly. Um, One of the things that I also see people struggle with is like thinking about experiments before they have a clear question or hypothesis. I think it's much easier if you sit down and think like, can I have somebody explain what my question is or what my hypothesis is very clearly? And then once you reach that point, then it's like, all right, what are the actual experiments that I'm going to need to be able to test this? I think one of the common things when you're early on in your career is to think very heavily on the methods before the big picture. So start thinking about like how can your hypothesis design your experiments rather than the other way around. And so if you struggle like, oh, what are the kinds of research questions I can do? It's this Venn diagram has helped me a lot. So there's an idea of what you want to do throughout the rest of your career and maybe what skills you need to achieve it. Like for example, if you want to go to industry and you know that data science skills are something that are going to help you get there, maybe you can come up with a project that overlaps your funded lab's goals and maybe their expertise, and also maybe like some papers that the lab published recently that intersect with your ability to gain data science skills, whether that's like computational programming skills, etc. Basically, you want to avoid a situation where, which I feel like sometimes happens, where like people kind of propose something that their lab is really good at, but then, the job search starts and then they realize that they might be really good at like, intracranial injections in mouse models, but then, maybe not a lot of people are hiring for that skill in industry. Um, or if you want to go into academic research, right? Think about like what are the kinds of things that you want to get out of that you might want to bring to a postdoctoral lab in the future. And if you can have preliminary data, whether, this is more applicable to like the grad students. If you have something in the lab, it could be something as simple as like a stain, histology, maybe a behavior that you ran really quickly, maybe an analysis that you did on an open data set that you're proposing to do with the data that you're collecting. I think having a figure that shows something related to like how your experiments are being set up already, how you're proactively working on this project already, that's even better in my opinion. Oh, and maybe this helps. If you do have preliminary data in the lab, it doesn't necessarily have to be collected by you, as long as you're like transparent about it, as long as you're like, oh, my lab's preliminary data shows blah blah blah, as opposed to like my preliminary data. I don't think people are going to judge you, especially in the first year of your grad school, if you don't have preliminary data that you yourself collected. So one of the things that kind of sort of in my mind when I'm evaluating a grant from one of my peers or one of my undergrads is like when I look at the overall hypothesis of the grant, I think about this checklist. Like one of the things that I see a lot people not be able to address really well is whether or not the hypothesis is actually going to be tested by your aims. And one of the other things I see people struggle with is making their hypothesis like very long, like almost like 3 sentences or 2 sentences long. Um, because and sometimes it's really hard to condense two really different specific aims into one single hypothesis because maybe they use different methods. But trust me, like if you sit down and think and zoom out what those aims are trying to address, you can come up with a thread that can be distilled into like a single overarching hypothesis. If you have um, maybe research that is much more exploratory, like maybe you want to develop, I don't know, like a connective atlas of amygdala to prefrontal cortex or something like that. If you know that your research is just kind of creating a database or even just putting out there like a resource, that is awesome. But I would highly recommend that even if you're going to do something exploratory, put in a hypothesis in there. Even if technically your data isn't restricted to one single hypothesis. Like, for example, if you do an atlas, you're not going to be like, oh, if I survey the entire brain, I'm only going to look at the amygdala. Um, like people understand that that's not why you did it, but nonetheless, it'll help you say that like, oh, to answer this question, we need to understand best, amygdala connectivity, and to do so, we're going to do a thorough, basically like atlas of nearby areas that might also coincide in terms of connectivity. Basically, people are going to understand that your aim is exploratory, but nonetheless, you're not going to be like caught by not having something that you're directly testing. And trust me, like even if it's exploratory, having something explicit in that aim will help the writing like a lot. Because then you don't have to explain all the things you have to do. You just have to explain like kind of like a funnel, like what the biggest approach is, and then get zoom in more and more and more until you answer that sub a specific hypothesis. And so one of the things that took me a while to understand is like what is a hypothesis versus what is a prediction. Um, in terms of the biomedical sciences, all you want to do is explain something in biology, right? And so the language that you might use, let's say if you care about how an area of the brain or two areas of the brain relate to a specific psychological phenomenon, then you are going to use like strong language, right? Like words like regulate or causes or correlates, um, versus like if you do a prediction, you're more describing what the result is. You're kind of zooming in a little more. So your hypothesis can be a little vague in a sense, maybe in terms of sometimes directionality, right? Like if you don't know whether two values are going to be like positively correlated or versus negatively correlated, you can still say that like they're correlated. And then in your prediction, you can say what direction they're going to be correlated under. And the reason for that is because you don't want to like if you fail, if your prediction is wrong, what are you going to do? Does your hypothesis still stand on its own? In your hypothesis, sorry, in your prediction, this is kind of where you then start saying like, okay, what about regulation of encoding reward between two brain regions? Well, it turns out that like maybe those two brain regions work to prevent that psychological phenomenon. Now you're talking about a specific effect. Now you're talking about like blockage or deletion of something, or when something might be attenuated, that is, something might decrease. Uh, um, whereas in the hypothesis, you're talking more about causation in a very sort of broad way. And then before you start writing, think about like what your experiment might actually look like in practice. This is why it's highly recommended that you propose things that you're familiar with or techniques that you're familiar with, because then you're not like kind of guessing. And if you get a reviewer that is an expert in that technique, then they're going to be like, well, this person clearly didn't address this thing, because maybe the only way you could have perceived that is by doing the experiment yourself, or maybe talking to somebody that has done the experiment in your lab. Sometimes a common pitfall I see is if your big claim is about causation, but your experiment really is only gonna like set up a correlation, um, pull back on your hypothesis. Like maybe don't say the word cause. Even if you know that like once you find a correlation, you're gonna do the experiments that are gonna tell you what causes it. People don't like it when you like confuse correlation and causation. So definitely pull back if you're only gonna do like uh, um, for example, if you want to say that like this brain region is important for this behavior, but all you're gonna do is record from the brain region while the animal's doing the behavior, you definitely can't say that that activity is causing the behavior, right? Because all you're doing is looking at a correlation. But if you propose like, oh, an optogenetic experiment, for example, then you can start getting more into like causality. Although people will still argue uh, whether it is necessary or sufficient. Um, and then people can get into debates about causality all the time in neuroscience. Um, and then one of the things that I was I felt unprepared for, honestly, like and I completely understand why, is because people don't teach us statistics often enough in uh, biomedical sciences, at least in my experience. Um, you want to be able to design your experiment in a way that like you're going to be sufficiently powered. So by that I mean that like especially at the NIH style grants now, they want you to be very like overt about whether or not you did like a power analysis or something like that, where you have a reason for the number of animals you're using, for example, or the number of subjects that you're going to recruit. Like you can't. find a large effect size that is believable if you only have like 10 subjects per group. Now, people that do rare disease research might run into this like roadblock. Again, you don't have to like couch your ambition. You maybe want to say that like you're gonna recruit enough people to be able to do a statistical effect that predicts that will show, a believable effect size that people have maybe shown through the literature already. If you don't want to do power analysis, one of the things, because that requires like some familiarity with statistics, although nowadays there's a lot of research online on how to do this. One of the easy things that people have done for a long time is just find a study that did something similar, show them or talk about their effect sizes or their statistics, and then say that you're going to do the same thing and that you expect the same effect or a similar effect, if not large. A large enough effect, sorry. And then kind of something that I see repeated over and over again about the specific aims, and maybe in the NSF-GRFP, literally your first page before you get into like the experimental design, etc., is that if you don't convince them that what you're doing is important and that you're prepared to do it, or that you have the environment to do it in, then you're not honestly going to convince them with the research strategy or with the training plan. Um, a specific aims page or like that first page of your NSF-GRFP research portion can definitely make or break your entire application. So this is if there's like a thing that you should spend the most time on are those parts of the application. Honestly, like if you have a solid specific aims page in the NIH style, the research strategy, which is 6 pages, just kind of flows because your research strategy is just like an expanded version of your specific aims. You're going to give them a much more thorough background. You're going to explain to them what each aim is going to do and what experimental strategy you're going to use. But now you have like 6 pages to do so. So even if you feel like you're not giving enough detail in the specific aims, that's okay. Because again, it's only 1 page. You can save that for the research strategy. The important thing about the specific aims is not like the technical details or the methodology. It's rather like what is the hypothesis? What is the question? What is the approach that you're going to use? And if it helps, it really is the page that people use to advocate for your grant in these panels. So if you were to think about like, can somebody just reading that 1 page be able to talk about my scientific problem, what gap there is, why is it important, and what are the goals, including like how I'm preparing to like achieve them. If you can sort of build all 4 of these, then you're well on your way to convincing them or to give them like good content for them to be able to defend your grant. And then because people read hundreds of these grants, often, early in the morning, maybe before the coffee kicks in, you want to make it as easy as possible for people to find the information. I do this a lot where I quite literally say the words. Our central hypothesis is, the objective of the proposal is, the rationale behind what I'm doing is, my expected outcomes are. I kind of leave it as easy as possible for people to look for these keywords like objectives, outcome, hypothesis, rationale, goal. I don't know. I think it works, to be honest, especially having been on like the other side, not, in a much lower stakes grant reviewing environment. I think this helps a lot because then I can just quite literally tell people what each of the components of your grants are. And keep in mind, people are not going to be experts in the exact same thing you do. So like they can't assume what your goal is. They can't assume what your objective is if they're not familiar with your field. So just tell them. And then some rules that I keep to myself when I write, especially nowadays that I'm a little more experienced, is like don't get like too sort of flowery. And I think nowadays with like AI assisted uh, um, writing, it can be very tricky to like, oh wow, this sounds incredibly convincing, and look how like academic this writing sounds. But people aren't necessarily going to be like open to academic writing when they're like reading hundreds of grants. If you can make it easy in terms of your language, then even better. And these are some like very straightforward examples that you might think are like, wow, this is common sense, but trust me, people kind of use often because they sound more academic. One of my biggest pet peeves is like people who use the word utilize when all you're trying to say is use. Again, because it's like nobody says utilize like in regular life, so it's kind of like, why would you use it in a grant if not to like make it sound more scientific? But the reality is that like maybe save that for the paper and not for your grant. Like even when you're giving a talk, you're not going to be saying like utilize or some of the more like jargon words. Okay, so we're good on time. I'm gonna check chat real quick. Yes, I will share this presentation after I am finished. That's the only question. And then I'll leave some time at the end um, for other questions. Okay, so now we reach the point where now we're going to talk about structure. Apologies to the NSF-GRFP people. This is not quite what the NSF-GRFP format is, but honestly, like knowing what I know now, I would just use it anyway. Especially, you can almost think of like the NIH specific aims page, which is one page, is like your research statement, except it's like almost more like one and a half or something like that. And also with the words, broader impacts and intellectual merit, more, overtly used. Um, so if I were to say like there are four components to a specific aims page, there's an introductory paragraph. There's like a second paragraph. I mean, I don't really have another word for that. And then there's your aims where you list them. And then there's like a summary paragraph. Once you understand the formula, it becomes a lot easier to write. So, thankfully, I have the biosciences writer website that I gave out has like a really nice formula that I really believe works. Um, so in your introductory paragraph, you want to start out with like people have told you this before, maybe like what is your attention grabbing statement? Honestly, like in the NIH style, one of the easiest ways to write an introductory sentence is look up like a statistic about something related to your research, whether that is like, if you do research in um, development, you might want to say like, oh, almost um, I forget what the statistic is, 8%, no, sorry, 3.5% of children in the United States have like autism or something like that. I could be wrong. You want to sort of hit them with a statistic. And the reason for that is because then you're literally starting out with like the public health impact or the public health relevance of your work. Um, One of the things you don't want to include in this paragraph is not just like that sentence that suggests why your work is important, but also like why is it not a solved issue? Like if you know that there's that many kids with autism and a lot of them, have comorbidities later in life, or that population keeps growing as diagnoses get better, like if that's an issue, a public health issue, because maybe the cost of taking care of individuals is going up, or maybe like the lives of parents are difficult as these children grow. Um, what is the thing that is preventing like sort of science from helping that issue? What is the blockade for to be able to answer like that large societal impact question? This is kind of where you have to be really creative about like identifying gaps, because maybe a lot of grants are going to say like, oh, XYZ is unknown, and that's fine. That's the gap. But one of the more effective versions that I've heard be used for this is like not just say why that something is unknown, but rather like say why it's unknown. An easy one is like, we don't know how this brain area is related to this behavior, so that's the unknown. But then if you hit them with like the reason we don't know that is because we don't have a technique, or we don't have a people haven't applied this like analytical approach, or people haven't used this like method from this other field on this issue. Um, the way I would think about it is like there's a gap, and people want to build a bridge to fill that gap, but then what stops people from building that bridge? And that is how you're going to address it, ideally. And then when you're coming up with these sentence or two, definitely align this. It goes back to what I said earlier. Align it to like the mission statement of the funding mechanism that you're talking about. Um, because honestly, like if you look up the mission statement of like maybe an NIH institute, they're literally going to tell you what the gap is in research in that field. And as I mentioned earlier, you want to start out with like your hook. And this hook should be very easy about what to determine like what your research topic is. So again, if you hit them with like, stroke prevalence is increasing by X% in the aging population, then it's very easy for me to think about like, oh, your grant is going to be about like either stroke or the mechanisms of stroke, or maybe about aging. I don't know, something that like immediately hits them with like what your research is going to be about. One of the things that I've learned, that I've also really stood by, is tell them what is known before you tell them anything about what is unknown. So like set up your stage before you are like, oh, however, XYZ remains unknown. And the reason for that is because if you sort of tell the story chronologically, it's a lot easier to absorb than jumping back and forth between like, we know this, but we don't know this. We know this, but we don't know this. Um, And then gap in knowledge I covered in the last slide, but the last sentence that I kind of highly recommend putting in this introduction paragraph is like convince them that there's a need to fill that gap. Whether that is like, another quick one is like the prevalence of this is increasing, or addressing this gap will allow us to like inform, diagnostics in the field that are much more personalized to the need of the patient. This is kind of like let's say like if your research were to be funded, what do things look like in practice like 10 years from now, for example? And I'm not going to read this entire paragraph, because you guys can read it in your own time once I send out the slides. But what I like about the resource, the biosciences specific aims resource, is that it quite literally like color codes different components of uh, um, um, what people look for in your specific aims page. One thing you'll notice here is that stylistically they do jump from known to unknown to known. Again, people are going to have different approaches to writing their specific aims page, but it does speak like you're not saying both things in the same sentence. Like they're quite clearly separated. And then like right now, in the need, people highlight in this particular example, people highlight the obstacle, like why hasn't this gap been filled? And then they just end with like why this is a necessary area of research. So now you've set the stage, what you're going to research, what is unknown, how are you going to fill the gap, or sorry, what is unknown and why it's necessary to fill that gap. Now you're going to tell them what you're actually going to start doing. What is your solution? Should be like kind of the first sentence of the second paragraph. Um, Convince the reader that like the solution is actually relevant to the gap. And you want to be, if I were to think of like what is necessary about this paragraph, is like what is it that you actually want to do, and why are you doing it? Maybe you want to tell them specifically like, oh, using this technique will allow us to fill the gap between X and Y that you mentioned in the first paragraph. And also like, how do you want to do it? This is a little bit of where you get into like the method, not quite explicitly, but like just generally what the approach is going to be. So for example, if you use EEG or fMRI, you might want to say the word neuroimaging. And then in your specific aims, you start getting into like the specific methods that you're going to use. And then again, this paragraph is going to vary depending on like the type of grant, or some of them are going to be some calls or grants are going to be a lot more about tool development, and that's okay. Some of them are going to be more hypothesis driven. So in this second paragraph, what I highly recommend are thinking about like what your own research goal is. So if your research goal is to fill a gap, well then make sure to mention that. Like my goal is to relate this to better be able to address uh, the thing that you mentioned in the first paragraph. I think a very central component of the second paragraph is like the words our overarching hypothesis is, or my central hypothesis is. Um, and again, even if you have like sub hypotheses in each of your aims, because maybe your aims use different techniques, keep in mind that like maybe those could be better framed as predictions or as working hypotheses. But your overall grant itself should be tethered around an overarching hypothesis. And then if you have the space, make sure that you mention like, or make it clear that what you're proposing is grounded in reality, right? Like you want to be able to cite a paper. Like for example, if somebody has shown that, you know, uh, just off the top of my head from my own experience, like, oh, people try to relate organoid activity to like EEG from children. Well, there's a paper from a couple years back that tried to do that, but they didn't do it. They did it in healthy children as opposed to children with autism. Like now you're setting the stage that what you're proposing is a logical next step in the field. And then another thing that is really emphasized when I took like a lot of training for grant writing is for training grants, your last sentence of your second paragraph should briefly state like why are you the ideal person to do this research. And let me show you what I mean by that. So here in this example, you're going to see color coded sections. The explicit goal is mentioned, what the objective of the proposal is, what rationale, that is what people have published before. Maybe people frame this as like just simply saying the feasibility of a proposed blah, blah, blah is. People want to know that what you're proposing is grounded in reality. Your hypothesis, this person wasn't super explicit. I recommend you are explicitly saying our hypothesis is, and then what the payoff is. So one thing I would replace here, so this is not for a training grant, by the way, this is more for like a PI grant. What I will replace the payoff with is like that sort of like, why are you the best person to do this sentence? One thing that I do is I quite literally say like my background in, electrophysiology, and then I cite the papers that I'm in, um, that show that I know electrophysiology, and then coupled by my mentor's expertise, and then cite some of the papers that show they're experts in that, ideally position me to conduct the following aims. And then you start with the aims. For a training grant, I think this is like really helpful for your reviewer to understand that like you're not coming in as a blank slate, and that you're in the right environment to be able to learn how to do that project. So the aims itself, people are going to have different formats for this, different ways of like how long they should be. Um, I think there's nonetheless still very critical components. So your aim title, I think, should explicitly, basically like, sorry, not explicitly, should heavily suggest what the goal is of that aim. And I'll go into an example in a bit. And importantly, your aims, I think, should be independent from one another. That is like, if you fail aim one, you should still be able to do aim two. And not basically like, again, think about it. Like, would you rather give like $200,000 to somebody that like if they failed immediately, where does that money go now? Versus somebody that's like, all right, like they're going to be able to move on and use that money to test this other thing. Um, Usually trainee grants are 2 to 3 aims. I really think that like my opinion is that 2 aims is good, because then it allows you to elaborate more about them. But some people are really ambitious and they have 3 and they still get awarded. So ask your PI what they think is best for you. And in terms of what specific is, right? Like you have your overarching hypothesis, like, oh, I want to elucidate something. I want to like correlate something. I want to like show the causation of something. Your specific aims is like now explicitly saying what you're going to show that supports that overall goal. So these are some examples that you can go through on your own, but immediately off the gate, you're going to be able to tell the difference in the language, right? Like this is a lot more general, a lot more uh, uh, user friendly for like general scientists. And then now when you get into your specific aims, that's kind of when you're starting to get into the meat of like what maybe model you're going to use, what gene you're going to investigate, what method you're going to use, etc. So by specific is like now you're getting into like the keywords. Maybe you're going to be using a little bit of jargon in your specific aims, but try to use jargon that is like already defined in your specific aims. Like it shouldn't be like defined in the specific aim what what ATGRP7 stands for example. So your title, I mentioned that it should be very clear what it is that you're going to be testing. One common pitfall I see is people using the word validate. Um, English is my second language, so for the longest time I thought validate meant something else and not what people whose English is the first language interpret as. Which I've learned, validate just means that you're showing something that already works again. That you're just doing it again, basically. I don't quite get it, but people interpret it that way. So you don't want to use validate because then it means that all you're doing is like confirming something as opposed to like doing novel research. And then in 2 to 3 sentences, you're gonna basically summarize what the approach is, what the method is, and what the expected outcome is. I'm not gonna lie, keeping your aim short is one of the biggest challenges, um, particularly because there's so much background sometimes that people spend introducing that by the time you get to your aims, you're like, oh my God, I don't have the space to say what I expect. This is kind of where editing and feedback is going to come into play. Like what is actually necessary in your background for people to be able to defend your grant. And then yeah, use headings or bullets to delineate each chain. So for example, this person doesn't use headings. I bold aim one, I bold aim two when I write my aims. You're gonna see your title, like you're gonna see strong words like establish, examine, and then you're gonna definitely mention what the experimental strategy is gonna be, what the approach is, and what the outcome is. So notice here that like there's less use of like the explicit, I will do this or I expect this. Again, this is stylistic choice. This person probably didn't have a lot of space. That's why they kept their aims really short. But again, different grants get funded. One thing that they don't have is rationale, like why or what has been done before that sets up this question or this specific aim. And then one thing that I've seen too is like people kind of having like their what I think of like the working hypothesis. So you have your overarching hypothesis, but now you're gonna get into like the hypothesis that actually is pertinent to the specific experiments you're going to do. I know we're out of time, but I just have like a couple minutes left. And that's mainly for the summary paragraph. So now by this point, the first time you write it, you're like already out of space. And that's okay. This is why multiple drafts are necessary. But I highly recommend you at least include like anywhere between 1 to 3 sentences at the end, like as its own paragraph that people can look at to be able to understand what the entire story is in a way. Um, you can think of it as like here you're restating maybe the title of your grant or like what the point of your grant is, but more importantly, what the impact of your research is going to be. Um, I think you're kind of zooming after starting very general and then zooming in into your specific aims. The summary paragraph is your space for you to zoom out again and be able to help orient the reader again outside of the tunnel vision that is a specific aim. And so maybe you might want to mention like, oh, this will be the first time that this is done. We will create a tool that the community will use, etc. maybe the outcomes in a much more broad setting, like, oh, these findings are going to provide like a molecular target to be able to test in like pharmacological experiments for this condition. And then your general impact, you're zooming out even more. Like, what is the broad impact of your research? One of the things could be like, well, you know, for example, like I study autism or I also study Rett syndrome. And when I talk about Rett syndrome, it's like such a low percentage of the population that has that condition. But when I talk about impact, I talk about it as like, well, understanding the mechanisms of this condition will provide a framework to be able to apply this to other conditions that are also like monogenic or neurodevelopmental disorders that have one single gene mutation. Because now you're thinking like very long term. It's like now people can be like, oh, I can take what this person did and maybe do the same experiments with Fragile X or Down syndrome, for example. Now you're zooming out a lot more. And this is typically how short they are, if not shorter. You talk about like, again, this will be the new mouse model. This will be the first time that something is shown. And from that, we're going to learn this thing if my grant is successful. And this is the large, zoom out 10 years from now, 15 years from now. Because you're successful, now the field has grown in this way. In a training grant, you also might want to like throw in here, like, if successful, not only will you be able to like do this for the field, but it will prepare you to be able to establish yourself as an independent investigator that studies XYZ. Like something as simple as that lets the reader know, like, okay, everything you're proposing is not only like a long-term vision for the research community, but also for yourself and for your career. And therefore, I want to fund you because I think you have potential to accomplish that goal. Cool. So now just to end off, um, your proposal, your call, whether it's internal, NSF-GRFP, or NIH, are going to vary whether or not they should include a specific games portion. But I really think like the format of the specific games really hits all the necessary components of what people are looking for in a grant. If you have a really strong specific games page, I'm not saying you should have a weak research strategy, but you know, it won't hurt you as much, is what I'm trying to say. And yeah, I mean, apply early. Don't give people your specific games page 1 week before their grant is due. Talking from experience, and then get enough perspectives from different people. And with that, so these are the different resources that I used to make this presentation. And I'm going to be sending out these slides, and I want to make a note for myself to like also send funding priorities examples for the NIH and the NSF. And with that, I mean, I know that I should have left more time for questions, but that's why I took pauses in between. But I'm also happy to stay on and like answer more questions, by the way, if people have them.
Thank you so much, Christian, for this very detailed and insightful presentation. A lot of profound information, guidance for anyone kind of, thinking about writing grants in the future. Um, these slides will be shared in the folder, in the workshop folder, in the G Drive. So you'll definitely, or the presentation will be there. And Christian, I'll also update you know, the folder with the slides themselves so that everyone can go in and click the links. If you don't mind, you could leave your email here so that attendees could email you with questions if that's something you'd be interested
Yeah,
in. Otherwise, I will stop the recording and I can leave um, I can leave the Zoom open for a couple more minutes so that you can answer some of these questions um, live as well. Well,
yeah, of
thank you
course.
everyone for being here, and thank you again, Christian, for the great presentation.
Of course. And again, you're not expected to know these things at